Welcome to Faculty Focus, the podcast of the Faculty of Pharmaceutical, Biomedical and Veterinary Sciences of the University of Antwerp, live from our annual Faculty Research Day, where we highlight the research of our professors and PhD researchers. I am Teun.
And I'm Marleen.
With our next guest on the podcast, we take a deep dive into our brains. Professor Emanuela Pasciuto is group leader for the Laboratory of Neuroimmunology at the VIB Center for Molecular Neurology. She researches the therapeutic potential of brain-resident T cells to reduce neuroinflammation. Welcome to the podcast, Professor.
Thank you.
Maybe a quite obvious first question, but what are T cells?
Well, T cells are a component of the adaptive immune system, together with B cell, for example, and they help us fight against infections and autoimmunity, for example.
Okay, and they're white blood cell, or?
They are white blood cells, yeah,
okay.
Indeed.
Okay, and before we go any further, it's maybe necessary to also tell us what regulatory T cells or Tregs or Tregs, how do you?
They are Tregs.
What are Tregs?
So Tregs are one subset of the T cells. So there are different subsets of T cells. So there are the CD8 and the CD4s. And amongst the CD4, you have effector T cells and regulatory T cells. So, and they have a very crucial difference. While the effector T cells are more known because they actually fight against infection and they are producing inflammatory cytokines, the regulatory T cells have the opposite function. So they actually stop inflammation and they prevent our body to react against our own body. So they prevent also autoimmunity. Okay. So their role is to stop excessive immune response.
Okay, and where do they come from?
So they have the same origin. They also are selected in the thymus, okay. Like normal CD4 T cells, but they are specifically selected for their ability to bind self-antigen, for example. So they have simply a different type of selection, but they have exact same origin as other T cells.
Okay, and the thymus is?
The thymus is most prominent in the children because during the first period of birth is where we develop our mature immune cells, while the cells are selected for their ability of not reacting against our body, except Tregs. And after the thymus gets shrinked, so it's very much reduced in adulthood, but it's still there, it's just very small.
And it's still producing T cells?
So the T cells are actually produced in the bone marrow and then they go through selection in the thymus. So yes, the T cells are produced throughout our life.
Okay.
Maybe to zoom out a little bit, why does our immune system sometimes overreact when staying calm would actually be better for our health?
So it's not actually like that. So the immune system needs to react and that's essential. So, but this response needs to start and needs to stop. So, and this is a physiological response. And this is why, so the Tregs are there to allow the response to stop at a certain point. And if this does not happen, then it goes wrong.
Okay. And you talk about the complex interaction between the immune system and the nervous system. Mm-hmm. But aren't all interactions between the immune system and the nervous system incredibly complex?
Yeah, indeed. This is true. So, but I think I found this especially complex because there are so few T cells in the brain. And I find so fascinating to see how just a few cells in one big tissue have such an impact. And this is because of specific property of the T cell. So I think it's a very specific challenge to understand as such rare cells can interact with so many neurons and influence them. So this is why I think it's especially complex.
And is it only are we just only beginning to understand how this system communicates?
Yeah, we really don't know much about it. So because so far we used, one challenge is that there are very little cells. So it's very challenging to study in detail their proteomic profile. So now we just limit our knowledge to transcriptomic because we have much more advances in single cell transcriptomic. But I think now our challenge is to try to really understand which protein do they express because they are the one that do the job. So they are the one that allow communication with other cells. So we are now moving towards more maybe genetic tools to study this interaction rather than relying just on single cell transcriptomic.
And could it be that some modern diseases are linked to pathways between organs or systems that we haven't fully explored yet?
Yeah, for sure. So, I mean, one example is, for example, autism, where we really didn't know that there was a role of T cells. So, actually, when I started my PhD in 2009, so I was working in autism model, and yeah, we really didn't know that the adaptive immune system could be involved at all. And I because I was so fascinated already at the time about the T cells and the adaptive immune system, I started looking everywhere, and I was so surprised that it was not done. And now, like just maybe a few weeks ago, a clinical trial was released in autism for modulating Tregs. So I think this is really a new era. We are learning so many new things about the adaptive immune system and the brain.
Okay, and on that note, you write that you broke the dogma of the brain immune privilege. What is that?
So, well, for many years, we thought that there were no immune cells infiltrating into the brain, specifically not adaptive immune cells. So we knew that there were just these resident cells like the microglia in the brain parenchyma, but we thought that it was completely sealed off from anything else, that nothing inflammatory could enter into the brain. And this is, I have to say, even though there are, besides my study, more publication already showing that, this is still, I feel, a bit a dogma in the field. So I really would like to spread the word and get this accepted, so that actually T cells are in the brain and they do not necessarily trigger inflammation. We just need to understand them better and know what they are.
Okay. And the levels of the protein EL2 can limit the number of T cells in the brain. Can you explain how this works?
So, well, it's not exactly this way. So basically, IL-2 is a cytokine which induces proliferation of T cells in general through binding a receptor that is called CD25. So, and this is especially true for the Tregs that especially rely on this cytokine to proliferate and survive. So now in the brain, this cytokine is very low. It has a very low level. So this is why we believe. So we perform some specific experiments. They are called parabiosis experiments, in which two mice are bound together, and you can study the kinetic of the cells. So what we can do is, because each mouse has a specific genotype, you can trace the cell. So for example, imagine a cell is always called A and B. You can always recognize in any moment A and B, and you can trace how a cell from one mouse goes to the brain of another mouse, and how do they change each other. So, and we use this model to calculate over time the kinetic of the T cells in the brain, because we had a time course. And we could see that the Tregs would just die very quickly. So while the other subset of T cells would accumulate in one other brain, not the Tregs. So, and we think that they are going to die. And this is because there is not much IL-2 into the brain.
Okay.
So you had also, you did also research where you delivered IL-2 boosts inside the brain.
We have developed a specific strategy in which we use a virus which only expresses into the brain. So this virus can be injected IV, so it's not invasive at all, but can reach the brain only. And then we get IL-2 expressed either by astrocytes or by neurons. It works very well in both cases, and then IL-2 is released very low dose in the brain only, not outside.
And it has an impact on which diseases?
So we studied so far the impact on traumatic brain injury in mice, and then also in stroke and in a mouse model of multiple sclerosis. And I'm currently studying the impact in Alzheimer's disease and autism.
Okay, and you had quite a positive result?
So for Alzheimer's it's going quite well, but for autism we just started, but I already know that there are also other groups working on it, and it seems very promising. So I'm very positive that it will work.
Okay. And yeah, we have to mention the Nobel Prize in Medicine, I think.
Yeah. I also mentioned it in my talk.
Okay. Because in the Nobel Prize in Medicine 2025 has recently awarded research in this area. Can you tell us a little bit more about that connection?
Yeah, well, so the Nobel Prize was given for the research in immune modulation, and I was very happy to see it. And there was one of the researcher that was they received the award was Sakaguchi. So that, I mean, when I started my studies in immunology, it was every review I read about Tregs was from him, because he's the one that discovered actually the genes that is associated to this population that was defined as immunosuppressive, that are the Tregs. And then the other two researchers were the one that actually discovered the syndrome. So this is the IPEX, because actually absence of this Treg is very dramatic and is actually lethal after a short time after birth. And this is called IPEX. This is where Tregs are missing. And the FOXP3 genes that is associated to the pathology is actually mutated.
Okay.
Yeah.
So you were a big fan?
Yes.
Yeah. Okay, that's great to hear.
Oh, just one final question. What advice would you give to a young aspiring researcher or scientist?
Yeah. So, well, I think something that I would advise is that they always have to follow their ideas and don't give up. And science is not easy, and answers are not simple. So this is a job that requires a lot of dedication and passion. And yeah, so they need to be really convinced and give themselves if they want to pursue. Otherwise, it's not going to work.
And find your own dogma to break, maybe.
Yes,
could also be.
Well, something that I would like to mention is that, is a bit generic. I'm really happy that I could continue these studies, because being able to link what I learned during my PhD on neurobiology and neuroscience to what I did during my postdoc in immunology is something I really wanted to do. And now research is very challenging. It's very tough to start a lab, and I'm so proud that I could do that here in Antwerp. And I found a very collaborative environment, a lot of input. So yeah, I really hope you can hear more from us in the future.
Yeah,
so
I'm sure we will.
Yeah.
Okay, thank you very much. And good luck with all your research.
Yeah, thanks.
Thank you.